जी, अस्सलाम वालेकुम student, welcome to biology online class. Dr. Fahad is here with us, and today Dr. Hamna is also here with us right now, and like people are also with us right now. As you know, last time we have gone through uh, initial chapters and their main target questions and some of their main techniques and procedures, how you are going to record your answer. A quick review up to 3 o'clock because other people will join with us. Uh, Whenever you are getting paper, MCQ, theory, or practical paper, try to go through the complete question first, read it, and then going to start the answer. Because if you start with, for example, it's a structure question and you are starting it and with part A, so maybe you write something of part B in part A. So in that case, you have to go through the question completely to avoid that issue. Another thing which they told me about कि आप start कर रहे हो तो आप दोबारा से student को introduction दे दो. So I just making a brief introduction as well. But right now, jab tak log start, they're coming to join us. I'm just waiting for the other people as well because we have 2-3 minutes left in this period of time. Brief introduction is this, as I told you last time, that I'm teaching here in Islamabad for the last 24-5 years. My name is Sarfraz Ahmed Qazi, and Alhamdulillah, my student they score A's and A stars. And some of the sessions in different institute, all the class they got A grade. It is unforgettable for me as well, specifically during the first decade of uh, this century. And one of the students, she got gold medal in the city school in 2003 in biology, as well as in 2007 regional distinction. Plus 2003 student, there are so many glimpses. I will show you. I will share you somewhere if it is possible. And Mavish Noor, she got a distinction certificate as well as a gold medal. They sent them. Plus, in every year, wherever I am taking the class, students are getting maximum A's, A stars. Very rare student are with B's or C grade. I think so. Negligible student they got D or any other grade. But overall, there was a 90-95% A's or A stars. As I told you last time, we have gone through about the time management of the paper. You have specific time for paper one. In GCE, you have 1 hour. For MCQs, you have 45 in case of IGCSE. MCQs, then structured question, 1 hour, 15 minutes, 6 questions. And in case of GCE, there is 1 hour, 45-minute question paper, structured question paper. And in case of practical paper, both of them, they are approximately, in case of GCE, it is 1 hour. And there are 3 questions that in paper 6. Same is the case for IGCSE paper. It is also round about 3 to sometimes 4 questions, but they are with, as I told you last time, some questions were related to magnification, some of them they're related to diagrams, labeling, and some of the questions, part of the question which are related to analysis, like osmosis, diffusion, potato strips, they are putting inside different concentration of hypertonic or hypotonic or isotonic solutions. And some questions are given from food test, as we have discussed yesterday, reducing sugar and non-reducing sugar test, plus Benedict solution test for protein, ethanol test for fats, and starch test, iodine test for starch. Although there is some question I have seen in the previous years, that is the vitamin C test, that is DCPIP, vitamin C, lemon or fruit juice, this lemon juice or orange juice is given to you, and you are going to add inside the drop by drop, or you are directly adding lemon juice inside the DCPIP solution, which is a colorful solution, it becomes colorless. More vitamin C, more quickly the color will be decolorized of DCPIP. So as far as your question, please be careful when you are drawing, labeling. Your labeling should be with the lead pencil, sharp pencil, straight line should be done. These parameters you have to do. inside that. Uh, at this time, I'm not suggesting anyone to go for the revision of 20 years paper or 10 years paper. I simply say just revise, excuse me, just revise uh, 3-4 years paper. If you have available, just solve it and give it to someone just to at least check it once. I will put today at least one solve check paper and according to the Cambridge of standard of Cambridge, and just to you can follow that as well. Now we are coming back to our today's lesson. That is where we stopped last time, and that was about coordination response. Now we are pursuing further. I'm closing the student. I'm closing the video because when I close this video, our speed of net is, I feel our speed of working is much better, much faster. That's why I'm closing it. Okay. Now, in addition to that, in case of chapter of coordination response, if you just recall, we have gone through neuron, sensory, relay, and motor neuron. In that case, so sensory, relay, motor neuron and their function. Sensory neurons takes impulses from sense organ to the spinal cord. In the same way, we have gone through function of some of the relay neuron. I'm taking you to the same topic where we were last time. I hope so you are here. Everybody is watching the slide. Please, anybody can confirm it. Students, hello? Are you watching the slide? The student, can you confirm? Anybody can confirm? Are you watching the slide? Reflex action, motor neuron, sensory neuron, relay neuron. Oh, thank you. Thank you so much. So nice of you. Thank you, Hassan, and thank you, everybody, those who have written here. Fahad as well. Okay, sensory neuron, relay neuron, motor neuron. Question on the basis of the same. Just look at the diagram shown here, and look at the question which was in the past paper, which they are going to have given you in the paper. I'm just putting one of the questions for the past paper directly, so we can be able to grab where we were last time. You can see on your group this question, which is directly coming in 2021. You can see this question on the group page. Everybody is watching this question. I have sent it on the group. Please just confirm. Do you have this question from the past paper? I have just uploaded on the group as well. The diagram shows. Anybody confirm it? The diagram shows the structure of involved in reflex action that result in the removal of person from the sharp object. So action that result in a removal of person's hand from the sharp object. आवाज नहीं आ रही. Oh, अच्छा. Just a minute. गड़बड़ कहाँ हो रही है? Please confirm. Are you watching the slide?
Yes, sir.
Oh, fantastic. I have uploaded one of the questions from the past paper, 2021. Are you watching that question as well, which is in the group right now?
Yes, sir.
Fantastic. Dr. Hamda, look at that. It's specifying in the question. Okay, what you are saying. Stuck the file and window. Stuck on the file and window. Are you watching the slide? Everything is clear, yes. Everybody? Some people are writing the messages about we are stuck in the... Okay, it's clear now. Thank you. Now, he's asking, the diagrams shows the structure involved in the reflex action that result in the removal of person from the sharp object. Just to take you this question directly on the paper as well, which I uploaded on the You are watching this question on the slide as well, on the screen, please. Are you here? Everybody watching the slide?
Yes,
Yes,
sir.
sir.
Excellent. Now, the diagram shows structure involved in reflex action that result in removal of person's hand from the sharp object. Remember, these questions are mostly coming anyhow from any angle. Any angle mean to say panoramically, they will give you any aspect of the question. Sense organ, what is the sense organ in this place? You will specify very clearly. For example, here, 5 senses. So one of them is here, skin with the touch or touch receptors. No, it is touching with the nail definitely. And sensory neuron take impulses, electrical impulses through sensory neuron, which is P, to the spinal cord, and Q, which is the relay neuron inside the spinal cord, inside the dark matter. Then R, it is the motor neuron, and which is ultimately ending in the muscles, which is S, and it is going to show contraction bending upward. So this is overall showing a reflex action in which brain is not involved. Now, as far as the questions are concerned, take a quick look. Structure P, state the function of structure P. Structure R, take impulses from the sense organ to the spinal cord P, and R, take impulses from the spinal cord to the effectors. That is structure R. You will also mention the name as well because he's asking name and state. Next, state the function of structure Q. Q is in the middle. That is the relay neuron or associated neuron or intermediate neuron. All 3 words are used in different books, so no need to worry Shari about this thing. It is a relay as well as intermediate as well as associated neuron. So these all words are for the middle one. So they are going to middle neuron, and they are going to interpret or translate the message according to the sensory neuron sending impulses. So its response are shown on the effectors, that is muscles or glands. In this case, it's a S is a muscle which is going to show the response. This is your brief answer which you will write according to the point 2 marks. Location Q in the spinal cord and all the brain also, because Q is inside the brain and spinal cord only, remember. And function wise, it interpret or translate the message. Now, this is about the neuron section which we had done last time. Now we are going further about just we are further going back to the next part that we wanted to start today, and that was about hormones, and which we have last time, which we are in the beginning of that just to start. So look at this one. Hormones, when we use the word, hormones are the chemicals or these are the biological catalyst, or we can say these are the biological secretions or chemical secretions, you can say. And these secretions are going to released by the ductless gland into the bloodstream, and they affect the target organ. So this is about the hormone in brief definition. So hormone definition should have few areas. 1, chemicals secreted by the ductless gland. 2, into the bloodstream. 3, and they affect the target organ. Remember this thing. So these are the definition of hormone. Now in the syllabus IGCSE and the syllabus GCE both, there are some of the hormones which are important. Just from that point, we are taking it a quick review of that. Take a look of this next slide where we are right now. I have taken one of the slide out for you. Now, endocrine glands, exocrine glands. Look at the both side as well. Endocrine glands without duct. Without duct, they release hormone, biological secretion, ductless gland into the bloodstream, affect the target organ. On the left side, I highlighted as well. Plus, if we go for exocrine gland, they are with duct, and exocrine glands include sweat duct, sperm duct, oviduct, pancreatic duct, mammalian duct, especially this mammary duct with the production of milk, and salivary glands. So these all ducts are, just a minute, these all ducts are exocrinal. So mean to say, when we say, because they are with the ducts, the secretions are released, right? This is the most important thing which you are going to be asked in the paper. I am also highlighting it to you as well and uploading it to you on your group as well. Now we are moving further to the next slide. Everybody is watching the gland slide now. Exocrine glands, just to confirm.
Yes, sir.
Okay. Sweat glands, salivary glands, mammary glands, stomach, liver. If you see on the right side, I highlighted again, pancreatic duct, because they release pancreatic amylase, lipase, and protease through duct. So it is exocrinal. Remember, testes, ovaries, they release testes release sperm, which is through duct. Ovaries release eggs, which is through duct, oviduct. So these are exocrinal. Remember. Now, as we move on to the next part, in the syllabus of both, there is some main areas related to pituitary gland, and which is the master gland, control all of the glands of the body. Pancreas, it produces two main hormones, insulin and glucagon. Thyroid gland produces thyroxine, control calcium, phosphorus level in the blood. Adrenal gland, very, very important. Look at that. Yesterday, we have done one of the questions relevant to that as well. Adrenal gland produces adrenaline, which is basically directly converting glucose from the muscles into the level of glucose which is inside the muscle. It is stored glycogen. It is converted into glucose. So glucose readily available for the time of flight situation. So adrenal gland produces adrenaline. I told you it has two types: adrenal cortex, adrenal medulla. Adrenal cortex produces corticoid hormone. Just to take a look of that as well. We are here directly on the adrenal gland. Yeah, adrenal gland, adrenal glands, and non-adrenal gland, adrenal medulla, and adrenal cortex. Adrenal cortex is outward, adrenal medulla are inward. So adrenal cortex produces corticoid hormone. Look at this one: glucocorticoid and mineralocorticoid, controlling glucose level metabolism of glucose, plus mineralocorticoid controlling sodium potassium level. But in case of adrenal medulla, as I told you, it controls. It is basically control glucose metabolism, specifically in a situation of fight, flight, happiness, sadness. For example, you got a news, you got A stars or distinction in biology. So you become extraordinarily happy, inshallah. So at that time, what happens? Or you got a news that Pakistan won a match or something like that. Any good news, which is your desire and your father is fulfilling it, or suddenly. So this, at that time, person become happy. So this adrenaline is released, and it is going to be at the time of anger, fear, fight, flight. What it does, they ask what is the function: increase glucose metabolism, increase cellular respiration, increase oxygen consumption, increase metabolism, because increase production of carbon dioxide. It also increase breathing rate, because when there is high carbon dioxide, there is high breathing rate. The body is going to get rid of carbon dioxide. Muscle contraction is also very fast. So heartbeat is increasing. So overall, its function is very, very important from your exam point of view. I'm also uploading it for you directly on your group. Now we are back. So these 7-8 functions you will specify: adrenal gland, increase oxygen consumption, glucose metabolism, heart rate, blood pressure, breathing rate, dilation of the pupil, increase carbon dioxide production at the time of fight, flight. Right, Hassan? Hassan Khan.
Sir Hassan.
Hassan Khan. Is it clear?
Yes.
Awesome. I would like you to quickly review what is the effect of adrenaline on the body. Just take 1 minute, quickly explain it, and when it is going to produce. Yes, over to you.
Sir, it increases pulse rate, makes the glycogen in the muscle get converted to glucose and also released in the blood. Sir, it increases concentration of glucose in the blood for respiration. Sir, adrenaline is, sir.
Yes, good, very good explanation. These are few points which are in front of you right now, and you can use that. Remember, adrenaline is also, after its effect, it is neutralized by non-adrenaline. Otherwise, its effects are tension, hypertension, hyperglycemia, raised metabolism, nervousness is there, headache is there. So this is basically related to adrenaline function, and your main questions which are coming in the papers, they are covering that areas. Now we are moving back to the you are watching the slide where we were last time. Our notes, LSE notes, please. Right? Are you there? Are you watching the slide of the notes, please?
Yes, sir.
Fantastic. Now, in case of LSE notes, if you see, the next topic, which important topic under that is effect of smoking. It's very, very important because drugs are the chemicals which are basically altering the nervous system by increasing the nerve impulses or by decreasing the nerve impulses, or giving the feeling of, we can say, euphoria or relaxation. These are the effects which are going to be. Just to make a quick review with respect to effect of smoking, because it is another important question which is coming in the paper and going to be asked. So as you know that smoking, mostly people smoke because of peer pressure, because of stress, tension, although it's contain nicotine, which is increasing the nerve impulses, and it has many side effects, although one advantage still there, that it increases nerve impulses, but it has 99% side effects. People are going to go for smoking for different reasons, just to show they're going to be chill or cool, or peer pressure, or a role model, something like that, or they're following him. Maybe the father is smoking or someone is smoking, then they're following them. It is a simply wastage of money, wastage of definitely energy, and leads to further diseases. Now, what are the cigarette smoke basically contain? It contains more than 4,000, 5,000, or some book says that 7,000 chemicals in cigarette smoke. Take a look of that. Very, very important. Nicotine. Nicotine in cigarette smoke, if you look at that on the right side, please, it stimulate the nerve neurons and increase nerve impulses and give alertness, increase adrenaline level, and also cause thrombosis, platelets aggregation, leads to coronary heart disease. Lumen size reduces, and it will lead to CHD, or if this blockage in the brain artery, it will result to the brain stroke. These are the effect of nicotine. Remember, in case of we can say cigarette smoke, first chemical, which is according to your book, or according to your exam importance wise, according to your exams. I'm just highlighting and saving it to you on the group as well. Second, look at that, the tar. Next one, it is a brown sticky substance accumulating in the bronchus, destroy the cilia, and mucus goes down in the lungs, accumulate in the lungs, and particularly this tar, brown sticky substance, it has cancer-causing chemicals inside that, many brown phenols, benzols, and all these chemicals, and many more. They will lead to a serious situation, cancer, tumor in the long run. So, and they also block the air passage, causes the cilia to paralyze, and they may cause bronchitis, inflammation of the bronchus, and lung cancer. Remember, nicotine goes into the bloodstream. Tar is outside the bloodstream, but it is overall, you can see, destroying the internal lining of the bronchus, cilia damage, and ultimately, because of the chemicals, they reach into the cell ultimately and destroy the chromosome by causing cancers, because cancer is uncontrolled cell division. Now, effect of carbon monoxide. It is again a third chemical which is too much in cigarette, and it is entering into the bloodstream. It has a great affinity for red blood cell, hemoglobin. It combines with hemoglobin, and it does not permit oxygen to combine with it. So it forms carboxyhemoglobin due to carbon monoxide. If level goes to 1%, then there is a great risk of atherosclerosis, mean fatty deposit inside the artery. So which will result into CHD, coronary heart disease. So fourth main chemical is irritants like hydrogen cyanide and acrolein, which paralyze the cilia and destroy the alveoli. People have a smoker's cough, or which to release the mucus out. They put pressure to release the mucus out. So the blobs of mucus goes out. In the same way, alveoli are ruptured and alveoli are not repaired. The disease due to alveolar rupture is known as alveolar damage, is emphysema, COPD, coronary obstructive pulmonary disease. So only solution of that is we should be smart. Remember, what is smart? Set a quit date for that. Tell the family friend we are quitting it from smoking. There is the only option we have. Then plan that from this, we are going to not going to take cigarette and remove cigarette from our areas wherever we are daily moving. Then talk to the doctors. You have physician consultant about okay, what should we do, because their effect starts coming out after many years, and person have to face in the long run a very hard situation. By the way, this is a briefing about cigarette and its chemicals. And now, as we move on, according to the syllabus, the next part of the chapter is most important, which is coming ahead: reproduction. In the topic of reproduction, please just to confirm that everybody is watching the slide of notes, like LSE notes, which we have right now.
Yes, sir.
Fantastic, wonderful. Now we are here with basically this is the most important drugs. All other drugs, alcohol, no doubt, they are all the other drugs important, but questions are based on that. If we put one by one each and every drug, so time scarcity is there. But just for your question, alcohol, it's a depressant, slow down nerve impulses. It has short-term effect. It has long-term effects, right? So alcohol is basically from that point of view. Although heroin is also there in the syllabus, they also it's a depressant drug. It is going to give the feeling of happiness. So affect the nerve impulses. So mainly questions which are based on the past paper, they are coming from smoking. Now reproduction. Reproduction is basically we can say two main types: asexual and sexual. Asexual, in which no involvement of gametes, no involvement of sperm and egg cell. Remember, so bacteria divides into fungi or fungi. They are producing back spores. They release spores, and spores are falling on the living or the dead matter as well. Parasite or a saprophyte. As a parasite or a saprophyte, they give rise to hypha, mycelium, and release enzyme, digest the food. And again, this process goes on. Hypha, mycelium, spores, sporangiophore. Fungus is going to grow without sexual involvement in this case. Another thing which is asexual method of reproduction, that is potatoes. We have seen on potatoes there are different eye buds. If you simply recall, if you see potatoes, it has some eye buds on it. कुछ इस किस्म के आपको नजर आएंगे. If you see some potatoes, it has some eye buds like that, or eyes or eye buds. When you cut the potato into pieces, for example, you cut the potato into 3-4 pieces, it should have 1-2 eye buds in it, and then you sow that piece of potato into the soil. When you sow this potato into the soil, for example, this piece you have taken, and you are putting it into the soil. It has 1 or 2 eye buds. It has stored food all around, give rise to roots, give rise to shoots, and ultimately leaves are built up. And due to photosynthesis, new potatoes are under the ground. Potato tubers are under the ground. They are building up in this way. So this is asexual method of reproduction. Mostly asked about asexual. Asexual method is going to give rise to identical new cells. Remember, identical to the parent organs in case of asexual. Now, as we go for sexual method of reproduction, it involves sperm and egg involvement. Sperm in the male and female and egg in the female, they fuse together and forming a zygote in the plants, in the animals, or human. Advantage wise, if you see, it produces genetically different offspring, so they don't all die, and there are more chances of in the environment for them. Disadvantage is that it takes a lot of time to definitely energize and good characters to sort out an energy providing process in case of pollination, as well as in case of if we go for animals or plants. So for the desired characters, it requires a lot of work to done, which we will do in the coming chapter. Now, right now, sexual reproduction in plant. They will give you a question directly of flower structure, and they will ask you the flower structure to label that. Particularly, the stalk of the flower is known as receptacle or thalamus, or the broad base. This receptacle holding 4 main whorls of flower. If you see these 4 whorls shown here, outer one is the sepals, then the petals, then there are stamen, and in the middle there is carpel. So these 4 whorls like that: outer one, calyx, then corolla, then there is androecium, and then there is gynoecium. So ask you to function-wise petals. Petal is going to be a colored part of the flowers mostly, and they are attracting insect for pollination. Sepals are green, which are going to protect the flower from hazards like temperature, wind, or moisture. They close the flower in the bud condition in the beginning, so their main function is protective from that point of view. Male and female part are the stamen. If you look at that, stamen has anther and filament. Anther has you see this is anther shown here. These are the anther shown here. If we make it little bigger, anther, it has, remember, for example, anther is shown like this mostly, and they have pollen sacs inside that. These pollen sacs contain male gametes, that is sperm, and this all anther is held by a stalk, and that stalk is known as this filament. So this collectively known as stamen. The male gamete is produced inside the pollen sacs. Remember, they are asking questions mostly on that. Okay, questions are coming where the male gamete is produced, or haploid cells are produced, or haploid cells are present inside the, you will specify in your here is n number of cells are produced, and these n number of cells, which is n number of cells mean to say the sperm cell, they are n number, or haploid, or you will specify in your answer they are haploid cells, these sperm and eggs. So with respect to just to clarify this areas, the female part, that is carpel or pistil, its other name is pistil as well. It has three parts: the stigma, the broad base, the broad tip, and a long stalk in the middle of stigma and the ovary. And this is the ovary, which containing ovule, and ovule contain egg cell, which egg cell is also with the n number of chromosome. Now here in this case, mostly they are asking a question about fertilization. Now, as far as the fertilization is concerned, it is the fusion of male gamete with the female gamete. Remember, just to make it little bigger, so we can make it little easier. I hope so. Everybody is watching the flower very closely.
Yes, sir.
This is a pollen grain which is landing on the stigma. It has sperms inside that. These sperm goes all the way by releasing enzymes, dissolving the tissues and reaching to the ovary, then into the ovule. Inside the ovule, there is a female gamete, which is an egg cell. And this egg cell, just to highlight here, which has n number of chromosome, or n number. Then it is fused with the sperm cell, which is also with n number. So when these two cell fuses, they form a zygote, and that is again 2n numbers. So in this way, the chromosome number of the species is stored as well. So this is fertilization, which is mostly asked in the paper to you people. Then in the Cambridge exam, they are putting a question like this. Just to show you one of the, I'm just also highlighting one of the questions related to this chapter on the group as well. After this fertilization, there is pollination. It is the transfer of pollen grain from anther to stigma. As you can see, the same diagram we are using. So transfer of pollen grain from anther to stigma, from here to here, or in this case, from anther to stigma, or from anther to stigma. This is self-pollination. If it is on the same flower, remember, or on the same plant. But if the transfer of pollen grain from anther, for example, this is one flower, and it is, for example, this is plant A, and this is another flower, and this is plant B, remember, and it has stigma, style, and ovary, and the pollen grain moves from here to the next plant on the stigma of the other plant, but of the same species. That is cross-pollination. Remember, it should not be missed or misinterpreted, because mostly the students they do mistake. If on the same flower, if on the same plant 100 flowers are there, self-pollination. If there are two plants of the same species and pollen grain is transferred, that is cross-pollination. And it is this cross-pollination is causing more chances of variation. Remember, more chances of variation. There are two main types of pollinated flowers which we have in the syllabus: insect pollinated flower, wind pollinated flower. Insect pollinated and wind pollinated flower, according to your syllabus, excuse me, insect pollinated flower are, they have colored petals, they have a small stamens, small stigma, but sticky stamens and stigmas. And another important thing, they have nectaries, nectarioids, and they are scented also. And in the wind pollinated flower, they have dull colored petals, they have no scent, no nectaries. And if you see this, for instance, I'm taking you to one of the slide right now, which is showing you, take a look please, about fertilization and then pollination. Gee, everybody is watching the,
Yes, sir.
fantastic. Now, when we say pollination, look at this one. The parts of flower shown here, they will ask you to label that, or they will ask you what is the function of each and every part, which we have recently discussed. Now, fertilization, they will ask you to show the movement of pollen grain down to the female egg. Then you can specify your diagram like this. Cell pollination, excuse me, cell pollination on the same flower or on the same plant. The pollen is transferred, pollen grain from anther to sigma, that is cell pollination. And if it is from one flower to the other flower, other plant of the same species, that is a cross pollination, as you can see here right now. This is one of the animation shown just to take you to another. There are some questions which I would like to ask you to share and tell me their answers quickly right now. Yes, starting from here, Hamna, question one. What is the name of the structure label X in the diagram? Yes.
Sir, stamen.
Stamen.
Correct. That's right. Very good. Yes, Fahad. Where is pollen made? Over to you. Dr. Fahad, where is pollen made? You have four options.
Anther.
Correct. Well done. Excellent. Gee, we have here with us, anybody would like to participate? Ishal is here with us. Yes, next question. Where is the ovule found in the flower? Ishal.
Ovary.
Correct. Excellent. Now, as we move on to the next one. Gee, we are here with Hassam Khan. Which part of the flower is labeled below? Yes.
Sir, X is stigma, Y is style, I guess.
Excellent. Perfectly right. Okay. Now, pollination we are discussing. We have discussed self-pollination, cross-pollination. Same flower, on the same flower transfer, on the other flower transfer, or the same other flower if it is transferred, and but of the same species or the other plant, it means it's a cross-pollination. So self and cross should be very much clear. Now, insect pollinated flower, as I was saying to you, they have small stamens and so small stigmas, sticky stamens and stigmas. plus they have colored petals, they have nectaries, they have scented. So these are the main characteristics shown here right now. And wind pollinated flower, they have long pendulous anther stamens, and they have feathery stigmas. You can see the diagram as well. So they are going to be quickly capture that from the wind. So they don't have nectaries, and they don't have scented, and they don't have bright colored flowers. These are the main characteristics of these type of wind pollinated flower, right? G, next question. We have here with us, और कौन है हमारे पास? अदील भी है हमारे पास. Ibra, Zahra. Irab Zahra is here with us. Irab Zahra. Well done. Yes, please. I would like you to participate here with this question. Pollination is the transfer of.
From the anther to the stigma.
Excellent. Yes, we move on to the other people as well. The two mechanisms of pollinations are, which we have recently done.
Insect and wind.
Correct. That's right. We have recently done is perfectly right. Okay. Cross pollination, what it does. Yes, anybody can participate. Cross pollination.
Sir, increases variation.
Perfect. No doubt it is increasing variation, because when one plant is crossing with the other plant of the same species, there are more chances of variation and new characters, new gene pool, new frequencies, and more adaptabilities. These parameters are going to be mostly inside that. Yes, next. Flower is adapted to wind pollination by.
Having feathery stigmas.
Excellent. They have feathery stigma, so they can be able to capture the pollen grains. Absolutely right. Now, this is a pollination. This is the mechanism of fertilization shown here. Anther to stigma. Transfer of pollen grain is it is showing here the pollen grain at the tip of the stigma, and it is releasing the male sperm into the stigma style ovary, the long tube, dissolving the wall by releasing enzyme and dissolving the tissues, ultimately reaching to the ovule where the egg cells are there. For example, if it is a maize seed, it has many sperm which is entering into it, many ovules. You know, maize, chalikadar, there are so many seeds. 100 to 1000 seeds are inside the comb, maize comb. So many sperms enter and fertilize with the sperm, and every sperm egg fertilization result into a seed formation. So this in case of plants. Now, as we move on,
I think we should explore a few solutions. Grammarly suggestions catch when your flower is pollinated and the pollen lands here, but it still isn't fertilization. The nucleus in the pollen still has to reach the nucleus in the egg down here. Now the pollen grows a long tube like a long straw, which funnels the nucleus to the egg. Seen under a microscope, these pollen grains are germinating. Pollen tubes are growing from the pollen grains. The male nucleus from the pollen passes down the tube on its way to the female egg cell, where they will fuse and fertilization will be complete.
Gee guys, this is about fertilization. Now we are moving back to our reproduction next next section, and that is related to germination. After, if you go on the next page, that is related to germination. Germination is basically the process from seed sowing to seedling formation. So it's a very, we can say, a step-by-step process. Now take a look of that. I'm sure everybody is watching the slide as well. Quickly confirm Please, everybody quickly confirm this thing that you are watching the slide.
Yes, sir.
Thank you so much. Now, germination, basically, if you see the seed structure, in case of, okay, just before germination, I would like to clarify one thing more, and it requires, there are some seed structures, sometimes they are asked to label that. It's a monopod seed. Seed had two main parts, remember? One is the endosperm, other is the embryo. Endosperm is basically containing food reserve, and embryo has three main parts: radicle, cotyledon, pluvule. Remember, in all the seed, there are two main parts. Outer is the covering, which is known as testa. Below testa, there is a shiny layer, tegmen, just below that. Then after that, there is embryo. And okay, no problem. So testa, tegmen, and in the same way, you can see the endosperm is a reserve food, which is whole the way inside, plus embryo. Embryo, if you look into the diagram shown here, a radicle, a cotyledon, or the pluvule. So this, sometimes they are asked you to label that. So try to be very much vigilant in this case, because this was the same diagram given in the past paper last year's, and somebody asked me to share that as well. Now it is on air. So two main parts. Which are the two parts? One is endosperm, second is embryo. Remember, so endosperm and embryo. Endosperm is the reserve food, and embryo is the three main part. Radicle, when the seeds start germinating, its testa break, radicle comes out first, then pluvule, cotyledon. Pluvule is going to form the shoot, cotyledon is forming the leaf. So first root by radicle, first leaf by pluvule, first stem by pluvule, and first leaf by cotyledon. Remember, just for your quick memorization, it is for the shoot or stem, or radicle will form the first root, remember, and the cotyledon will form the first leaf as well. So these are the main introductory area of the seed structure. Now we are going back to as we are discussing about seeds, and seeds have two main parts, that endosperm and embryo. So this we have just recently gone through. Now seed have an outer coat, that is testa, then there is radicle, pluvule, cotyledon. There are two types of main seeds, as you know. One is monocot, other is dicot. Monocot seed, dicot seed, basically wheat, rice, maize, grasses, they have monocot seed. And dicot seed, in your syllabus they specify some gram seed in detail, or bean seed in detail. So these are the dicot seed. Excuse me. This is the overall structure: monocot at the bottom and dicot at the top, with the complete labeling. Just to the same thing which we have discussed. Remember, there is nothing new in it. All the two main parts, and the two main parts are, as I told you, and they are embryo and the endosperm. Embryo has three parts: radicle, pluvule, cotyledon, and endosperm is a reserve food in both the cases. So when water enters, seed become activated. Now here, this is seed germination, and radicle comes out first, which form the first root. Pluvule form the first shoot. Cotyledon form the first leaf. Now we are going back. Next part is of this chapter is dispersal. Again, dispersal of seed and fruit. Now there are four. Look at that. There are basically dispersal. Basically, it is the transfer of seed and fruit from one place to the other through different agencies, like wind, like water, like animal, like explosive mechanism. So these are examples shown here. In case of wind, we have 3-4 examples: dandelion and angsana, shoria. These are the 3-4 examples. Mostly they are given. And exer, these are the plants which are with the wing-like structure, parachute-like structure. So they move from place to place. Excuse me. Next is animal disperse. They have some spine or hook-like structure which attaches with the fur. Animal move from place to place. They also transfer seed and fruit in such a way, and from one place to the other. Then animal disperse, as I'm saying to you, sometimes animal eat the fruit and move from field to field, and within 1, 2, 3, 4 kilometers away, or many kilometers sometimes. Some, cows, goats, sheep, camels, even they travel all the day long, 10, 20 kilometers even, and they spread the seed after eating and doing dungs in some other place, or feces or excretion. Seed germination, as far as is concerned, seed germination, remember, student, they do a sometimes very big mistake. Germination of seed require 3 main factors. Remember, 1, temperature for enzymatic activity, 2nd, oxygen for respiration, and water as a best solvent for all the metabolic activities. They will give you a question based on that. Anything, design an experiment and do with explain with describe investigation. Temperature is necessary for seed germination. So you are going to specify the apparatus, method, procedure. In that case, you can take, so for example, just to take you somewhere with respect to this investigation. Gee, everybody's watching seed germination. Please just confirm it. Are you watching the slide of seed germination?
A bean is a type of seed.
Yes, sir.
It has a protective coat, and inside is a food store. Here are several beans which were left to germinate on water-soaked paper.
Okay, how the question come? How we will handle that? They will ask you effect of temperature, effect of water. So you can take, for example, for three petri dishes, putting same amount of cotton wool, same amount of water, same type of seeds, for example, bean seeds, soak them a day before. Then you are putting same amount of number of seed in all the petri dishes with which you are working. For example, 20 seed in one petri dish, 20 in the other, 20 in the third. If you are testing for temperature, and place them, one is in at for example at 10 degrees C, second petri dish with the all the condition fulfilling at 20, at third in the 40 degrees C, at the and one you will place more than that. So you will see the seed germination. In case of 10, it will be slow, less seed germinated will be germinated. 20, it will be more. At 40, 35, 40, it will be maximum, optimal. And above, it will be less. So in this way, you will write at the end some safety precautions. Remember, always write safety precaution in every practical like this. Goggles, gloves, hair tie back, tie clips, lab coats. And one more thing about reliability or accuracy. Repeat the experiment 3 times. Record the readings, write their means. These parameters you should mention in your accuracy or reliability section. Right? And finally, remember, also mention the conclusion as well. Okay, if it is related to temperature or water, or it is related to oxygen.
This bean has started to germinate. The first root has grown. This is called the radicle. This part will become the stem. It's called the hypocotyl. This bean was germinated upside down. The radicle grew up and then down. The hypocotyl grew downwards initially and then grew upwards.
Now, I'm uploading one of the past paper questions solved diagram. Take a look on your group of LAC about the dispersal dispersal of seed. Everybody is watching the question. Yes, please. Can you quickly mention? Everybody is watching the question, please. The student, are you watching the question, please? Hello? The students, are you hearing my voice? Are you watching the question?
Yes, sir.
Okay, look at that. A diagram shows PQR, P, Q, and R. With reference to the structure shown in the diagram, how each fruit is dispersed. Look at P. It is dispersed by animal. It has spines and hook-like structure, so by which it can be easily attached with the fur of the animal, and the animal move from place to place. Second, Q. It is dispersed by animal. This second one, it is also dispersed by animal because it is eaten by the animal, and seeds are not digested, and they are going to be released out during feces or during egestion. Then lastly, it is a wind dispersed seed. Take a look, and it has a parachute-like structure, dandelion. This is the dandelion seed, which is R, dandelion fruited seed. Fruit is at the bottom, and modification, you can see a parachute-like structure. So it can easily float in the air from one place to the other. Now, sometimes they are putting a question: what is the importance of fruit seed dispersal? If all the seed is going to fall on the same place, they will increase competition for resources like water, mineral, as well as light. So it avoid competition, number one. Number two, on the new place, they are going to colonize. So there is more variation. There is more chances of resources available. And it also minimizes competition. More availability of resources like light, water, soil. These parameters you can specify in your answer as far as the advantages of dispersal is concerned. Okay. Now we are back here with our own slide where we were last time. We are here with reproduction in human. As far as the human reproduction is concerned, there are again 3-4 important areas. Male-female reproductive systems are concerned. Their labeling is going to be asked, and then afterwards labeling with respect to different organs. Sometimes they are asking where the sperms are produced inside the testes, haploid number of chromosome, and they will ask sometimes what is the role of how the sperm is nourished. In that case, you will specify the glands which are all around, the seminal vesicle, the prostate gland, the Cowper's gland. They are going to nourish the sperm. Plus, in case of female reproductive system, they will also specify where ovulation takes place, where fertilization takes place. I'm just putting some process on that. Ovulation takes place here. Egg release from the ovary at day 14 of every female in the human, because we have human reproductive system. That's why in the syllabus. Next, right ovary release 1 egg and left ovary release 1 egg per month. So alternatively they produce. So 6 eggs released on the right ovary 6 months, and 6 eggs released from the left ovary alternatively. So 12 eggs are released from this place. Second, where the fertilization done, egg stays inside the oviduct, where I'm putting the next cross. Egg stays in the oviduct here. Here fertilization takes place, and as I told you, ovulation takes place here in the ovaries. Second, implantation takes place. Just to make the change the color so you can be able to grab the thing here. Implantation in the uterus. There are three places where implantation can be done: here, here, or here. Right side, left side, or on the base. So this is implantation area. Just to put I for implantation for your memorization. IMP, implantation. Okay. Now next is very important area, mostly coming in the paper, like what is the role of hormones in the menstrual cycle? If you just see this closely, just to make it a little bigger in size. Role of hormones. If you see the first, there is uterus lining thickness is shown here. First 5 days, it has gone down. It has gone down. Remember, it has gone down. And after 5-6 days onward, up to day 27, 28, it goes up to the peak. Then again, it goes down. The next cycle starts from here onward. Now, pituitary gland produces two main hormones. That is FSH and LH. Remember, FSH go a little high, then down, then high, and then down. Remember, LH is again it high nearly 12, 13, 14 day and go to the peak on day 14. It comes down again on 15, 16 and again goes back. This is LH shown. Ovary produces two main hormones: estrogen, progesterone. Estrogen increases in the first few days and continuously increases up to day 11, 12. Remember, day 12, 13, they go down and egg goes out on day 14. So before that, so it helps in, estrogen helps in ovulation. Remember, number one. Number two, estrogen also helps in follicle to convert into Graafian follicle. Immature egg into mature egg. Remember, this is follicle and this is a mature follicle. This is follicle, immature egg. Graafian follicle is a mature egg. Next, the remaining structure which is left over after ovulation is corpus luteum. Now you can see estrogen level little goes down, then high, then down. But progesterone level on day 12, 13, 14 continuously rises, and again goes down on day 28. Main purpose of the progesterone here to make the uterus lining thicker. Remember, if fertilization takes place, remember the progesterone level remains constant up to 9 months. 9 months, it remains constant. Remember, and make the uterus lining thicker for the implantation of the embryo. And after implantation, definitely it is continuously released in the bloodstream, that making the thickness of the uterus. When there is no fertilization, remember what happens? The progesterone level falls, which is detected by the pituitary gland. Take a look. Here the progesterone level falls. This progesterone level is falling here. It is falling here on day 28. Then pituitary gland sense it that the level has fallen down. Then they start producing FSH again. Pituitary gland start producing FSH again, and the new follicle becoming a Graafian follicle. And this cycle ultimately next goes on. Now, in shortcut, you have to memorize FOLP. Remember, this is the shortcut way. What is this shortcut way? Take a look. Follicle stimulating hormone triggers ovary to produce estrogen. Remember, estrogen, after its ovulation, it triggers the pituitary gland to produce LH. LH triggers the ovary to produce progesterone after ovulation to make the uterus lining thicker. And then, when there is no fertilization at day 28, progesterone level falls down, a feedback mechanism starts, and again pituitary gland starts producing FSH. So it is a shortcut to memorize FOLP. Remember, as we move on, after that, there are some other parameters in this chapter. Before we enter into the next parameter, I would like to share the past paper question here right now at the spot, because it's recently this question was in the paper. Take a look of this question. Are you watching the question here right now?
Yes, sir.
Fantastic, thank you. No, this was question which we have done yesterday. Just after that question, they are putting this question. Take a look. A and B. He's asking this. You have this in your notes as well. LSE resource pack. The blood concentration of two hormones A and B released by the ovaries during the menstrual cycle. The thickness of the lining of the uterus. Now, release the blood concentration of the hormone shown here. Secondly, 0 to 28 days mentioned very clearly. And here you can see the thickness of uterus shown in the next graph. Now, as we move to the question afterwards, describe the describe the roles in the menstrual cycle of hormone A. Hormone A is estrogen. As I told you, it is triggered by FSH. It is triggered by FSH, and this A is estrogen, which is produced by ovaries. Its function to make the follicle into Graafian follicle, and also helps in, and also it does ovulation, helps in ovulation. At day 14, eggs release out. Then it level falls. So this is about the function. State the day at which ovulation most likely to occur. Day 14, here. Remember this day, day 14. It is ovulation. Egg goes out. If the intercourse is done, fertilization is confirmed. Next is, if we go for the next part, next question, what was that here? At which day, when the lining of the uterus is lost from the body. So these are the 5-6 days approximately of the menstrual cycle from here to here. From day 0 to day 5-6 approximately. So menstruation takes place here. You can see the endometrial lining, it's washed off, it's ruptured, and bleeding takes place. So these are the questions based on this menstrual cycle. And now we move to the next important part of this chapter, relevant to where we were last time. Just to go back, clarify this whole. We were on page number, just a minute. So in the case of this chapter, there is a last part which is more sometimes is important related to method of birth control. In that we are discussing it right now, some of the parameters. Then we will move further to the next chapter. By the way, method of birth control mean to say there is family planning. So family planning issues, just a minute. It should be in front of you. Then we'll move further. There are different methods by which the family planning can be done, or method of birth control are used for that. Here. These other parameters are very easy: breastfeed, bottle feed, and developmental stages. I'm directly moving to the method of birth control. Look at that. The natural method, it is if the couple is going to go for intercourse between day 5 to day 11, there is 99% chance no fertility, and from day 17 or 18 to 28, again there is 99% chance there is no fertility. That is natural method of birth control. So female knows the days of the menstrual cycle according to that. Second, chemical methods. Chemical methods of birth control, two main chemicals in the syllabus. One, contraceptive pills. They contain estrogen, progesterone, artificial inside that, which is continuously making the in the blood level of progesterone high. That's why FSH is not triggered and no follicle become a Graafian follicle. Spermicides, chemicals are available in the condoms and in the feminoms, and creams are used which are killing the sperms. So spermicide for killing the male sperms. Mechanical method, in this case, there are two main devices which are used. One is that is a T-shaped device or the spring shade device, and also condoms and feminidoms are used to protect the sexual attraction or to avoid the chances of baby. There is another important method which is not mentioned here, that is which I mentioned here. Yeah, yes, that was I was just not surgical method. Look at that. That is vasectomy. Very important. Remember, the vasectomy. Vas deferens is cut and dyed. Means sperm duct is cut and dyed. And in female, the oviduct is cut and dyed, which is known as tubal ligation. So, female sterilization or tubal ligation. So these two methods gives 100% control as far as the birth is concerned. Next is, we are here with the inheritance. In case of inheritance, I would like to share with you very, very important areas today. Chromosome structure. Remember, chromosome is made up of genes, and chromosome is a double helix, is a ladder-like structure, which is made up of A, T, G, C sequences. In this chapter, we will study the main definitions like haploid, diploid, half number of chromosome, gamete cells, full number of chromosome, or diploid number of chromosome in the body cells. So these chromosome numbers, they are very important as far as the mitosis and meiosis is concerned. If you go for mitosis, slow. AC को slow करें. सर पे हो आ रही. So nice of you. AC का number जो है ना वो उसको 16 पे है. Please put it on 20, 21 or 22, 3 पे कर दे. All the cold air is blowing on my head directly. Thank you. Okay, students, we are back here. Everybody is watching the slide. Please confirm.
Yes, sir.
Fantastic. Okay. Now, mitosis, meiosis. Sometimes they ask about difference between. Thank you so much. Mitosis, meiosis. Mitosis is a type of cell division in which the chromosome number remains constant. Specifically, and in meiosis, the chromosome number reduced to half. In short, they are asking you what is the significance of mitosis, what is the significance of meiosis. So mitosis takes place in the somatic cells, meiosis takes place in the gamete cells. Mitosis produces identical new cells which are like the parents, and meiosis produces new cells which are different from the parent, although the number of chromosomes is half, or one division by meiosis result into 4 cells formation, and they are genetically different from one another. Take a look of mitosis, please. Mitosis, you see chromosome shorter, thicker. Then number 2, replication of chromosome takes place, 46 plus 46, and ultimately chromosome align themselves on the equator. You see this is the equator shown. Okay. Oh, yeah, no problem. Okay. Those people who are in need of a little break of 5 minutes or so, please, you can do your quick. You want to go to some change or some want to go to the washroom, you can go for that. I will recap once again. No problem. That area, right? Or I will, in this period of time, if somebody is in need of some break for a few minutes, for going to the washroom or something like that, so you can go. I will repeat from the same place where you are. No issue. Now, chromosome shorter, thicker, replication is done, chromosome align themselves with the equator, and chromosomes start pulling apart. These chromosomes become chromatid and they start pulling apart, and ultimately 2 daughter cells are forming with the equal number of chromosomes. No, this is about mitosis in every cell which is taking place, remember. And this is also showing some role. When we say its importance, it's very, very important thing here right now, that what is the benefit? What is the significance of mitosis? It helps in the new cell formation, for the growth, for replacement of the damaged cells, injured cells, and ensure exact copy of the parent cell is formed. Asexual reproduction is also a very important area. In potatoes, we have gone through. In fungus, we have gone through. In bacteria, asexual reproduction is done. But right now here, remember, when we use the word asexual reproduction, that is related to potatoes or animals or plants, wherever there is. If there is any, in case of asexual reproduction, there is no involvement of gamete, as I told you, like potatoes, like ginger, like onion, they produce asexually. They have some buds on it and they release roots downward, stem upward. So this is for growth, asexual reproduction. Asexual for exact copy of the parent cell, identical to the parent cell. Desired characters, whatever they want, it is also going to be easily tackled from that. Next, cloning of the tissue. Cloning of the tissue, like human organs, heart, kidney, lung, liver, and other tissues, muscle tissues, they are also cloned by a single cell and grown in the lab inside the laboratory, and ultimately they are going to make an exact copy of the parent cell. Now, as far as the meiosis is concerned, remember meiosis is a reduction of DNA. Look at that, the stages shown here. The upper one cell divides into two. Excuse me. (46 to 23, and these 23 cells, 23 chromosome. In the next division, they are going to form 4 cells, and again you can see the 4 new cells which are forming. They are with the haploid number of chromosomes. So in the beginning there were 46, and at the end 4 daughter cells with haploid number of chromosomes in case of sperm, in case of eggs. Remember, so this is about meiosis, which is taking place in our body, right? What is the significance? Question come to you. What is the significance of, we can say, meiosis? So in that case, if you are going to be asked, so that what is the significance of meiosis? Meiosis is a reduction division. It causes variation, right? It produces gametes, male gametes and female gametes. It shows genetic variation in the new species. The daughters are not exactly like the mothers. The sons are not exactly like the fathers, just like that. So this is the new plants which are produced from the parent plant due to meiosis. They are not exactly like the parent plant. So but due to meiosis, another plus point is that advantage is this, that it restores the chromosome number of the species. Remember, so these are the few areas which are related to mitosis and meiosis. We are back here with monohybrid cross. Very, very important area of this chapter. Monohybrid cross. Remember, if we say monohybrid or dihybrid cross, monohybrid cross means one character is taken and it is crossed. For instance, there is I'm taking you to some slide. It will be more easy to understand that. It's okay. Thank you so much. Gee, everybody is watching the new slide, just to confirm that.
Yes, sir.
Okay. Gee, are you watching the slide of monohybrid cross?
Yes, sir.
Yes.
Thank you. Thank you so much. Monohybrid cross means single character is crossed. You know, there are millions of characters in the male and female. For example, father and mother. In the same way, plants, the male plant, the female plant, they have millions of characters. We are selecting only one. For example, take a look of this diagram shown here. A tall plant and a short plant. A tall plant, for example, it's a pea plant which is tall, and it's a pea plant which is short. We are representing their gene. Remember, genes are the information of the chromosome. It is a part of DNA on the chromosome. And it is represented, we are representing the gene in the form of capital or small letters. If they are written in the form of, for example, this is, remember, this is a gene. This is another example of gene. This one, T small. So these are the genes shown here. When the genes are, remember, genes are always in pair. It's very, very important. Genes are always in pair. When they are in pair, we call them I'm writing it short, homozygous dominant. But if they are capital small, we call them heterozygous. Similar genes are known as homozygous dominant genes. It can be similar for tallness. It can be similar for shortness. Remember, it is HOR. So, take another example on the right side. For example, red. Red color genes are here, RAD shown. For example, white color genes with RR small, white. So, in this way, you will specify that genes are always in pairs, and R capital, these are the genotype. When we say, what is genotype? Represented with the letters. This is genotype. If we see the external appearance, that is known as phenotype, red color. RR small is the genotype for a phenotype, white. So genetic makeup, excuse me, and phenotype is the external appearance. Remember, just to show this example, to make it a little smaller, so all the slide can be clear in front of you. Now we are discussing the area, a tall plant with the dominating gene, T capital, and that is, I am writing with them as well. It is homozygous dominant. Remember, and I'm also writing with the short one. It is homozygous recessive. Now, when they are crossed, all the first generation which is formed, that is HE, heterozygous, 100%. Excuse me. 100%. Remember, with out of 100%, we take two individuals. Out of these 100%, we take only two individuals, heterozygous individuals, and we are making a cross of that. These are heterozygous both. So when we will make a cross of that, we will get TT capital. You can make for your convenience, this like that. You can make a Punnett square like this. T capital, T small. T capital, T small. Now here, if we make this, the uppercase is the, for example, the male side, and the lower one is the female side here. For instance, now our first probability is T capital, definitely T capital T small, and then we have the next possibility is T capital T small, and the last probability is T small. So we wrote it here for you. These are the genotypes, and their phenotypes are tall, and 25% small. Look at this one. Tall. But these two are the carriers. Remember, these two are the carriers of the shorter genes. I'm putting a block on them, and they are 50%. And lastly, just to change the color for that, it's a recessive one, the short one. So we have very, very important. We have phenotypic ratio 3:1. 75% tall, 25% small. But we have 1:1, 1:2:1, the genotypic ratio. Very, very important. Look at the genotypic ratio. 25% HOD, 50% HE, and 25% HOR, homozygous recessive. So our genotypic ratio is, just to write in front of green, genotype, and the lower one is phenotype. So genotypic ratio is 1:2:1. Phenotypic ratio is 3:1. Remember, it is the best example always coming in the paper to you, anyhow, related to the topic of monohybrid cross. I'm putting it also here on your directly the group as well. Now, next, as we move on, very important area. Some of the characters, they are in the body, they are dominating characters. Some are recessive. Did you see? Tallness is dominant over shortness. Black hair is dominant over brown. Curly hair is dominant over straight. Black eyes dominant over brown, or brown is dominant over blue. Hanging earlobe is dominant over fixed earlobe. Roller is dominant over no roller. Tester is dominant over no tester. Dimples are dominant over non-dimples. Normal vision is dominant over color vision, color blind vision. Then skin pigments. Skin has no pigmentation. Albino peoples, they are recessive. So these are the few areas which are. Now there are two short questions which are I'm putting it to you, and I'm giving you 2-3 minutes, please. Make a Punnett square and put it on the group. Time start. Question 1, question 2. Students, you have 2 minutes, please. Time start. It's a monohybrid cross. Very good. Hurry up. Everybody is working, please. Yes, which of the following crosses is likely to produce 3 brown and 1 blue eye? Make a correct cross, a Punnett square, and send your snapshot on the group. Time start. Hurry up. Question 2. Which of the following crosses is likely to produce the ratio 1 brown and 1 blue? Student, is this question clear to you? Everybody? Ji, Hamna, are you watching the slide right now of the question?
Yes, sir.
Okay, Hamna, aapne karna kya? It's very easy. Just to make a peanut square like this. Okay, Hassan, ऐसे peanut square banana just to make a peanut square like that. And he's asking 3 brown or 1 blue कैसे होगा? What will be the most closest option? आपने सारों को try करना है. One by one. इसको थोड़ा सा we will use directly on the board. Take a look. यहीं पे करते हैं. अभी करते हैं. फौरन. Simple mathematics. इसमें क्या difficulty है? Take a look. I am doing for the first option. Make the peanut square. Okay, we take some other color. I think so. Blue is better. For the first one, please. I am doing the first one. B capital, B capital. Second, B small, B small. We are getting here B capital, B small, B capital, B small, B capital, B small, B capital, B small. So it is not possible because he's saying that to produce a ratio of 3:1, yet 100% all of them they are brown. Agree? So it is not possible. Agree? One is not correct. Right, Hassan? Right, Fahad?
Yes, sir.
Now we are moving for the next one. Next question. For the next question, what we are doing, heterozygous B capital, B small, B small, B small. Now we are making a cross, B capital small, B small, B small, B capital, B small, B small, B small. This cross is also not correct because it is giving us 50-50% ratio, 50% brown, 50% blue. So it is not correct also. So third one, we are using the third option, and that is B capital, B capital, B capital, B small. So here is B capital, 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 B capital, B small, B capital, B small. So it is giving us 100% again. So it is not correct option. Last we are trying B capital, B small, B capital, B small. So here, just to make it later. B capital, B small, B capital, B small. Here is B capital, capital, B capital, B small, B capital, B small, B small, B small. This is the right option. Look at that. Option D is the correct option. 3:1. These three, capital, 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 small, 75%. As mentioned here, 3 brown and 1 blue. Gee, what is the question? Correct. Well done. Excellent, Fahad. Okay. Now move to the next question. Please, guys, you are getting 1 more minute and do it. Time start. For question 2, I have done 4 options for the first one for you. You can do the same thing quickly and tell me which is the correct for that. Question number 2. Which cross is likely to produce ratio of 1 brown and 1 blue? आपने सबका you will make the peanut square for all. Remember, आप सबके लिए peanut square बनाएंगे ऐसे करके, and then you will write the correct one. Remember.
Sir, is it option B?
Option B, correct. That's right. Well done, Hamna. Heterozygous with the, okay, we can go for this one as Hamna has done. B capital, B small with B small, B small. And we are here with B capital, B small, B small, B small, B capital, B small, B small, B small. Very good. It is 1:1. So this is 1:1. Perfectly right. Okay, Ji. Now we are moving to the next question. Yes, question number 3, 4. I'm giving you 2 more minutes, please. Then we will move to the next part of this chapter. Practice makes the man perfect and practice makes the woman pure. Hurry up, guys. Speed up yourself. Take 2 minutes to find
Sir,
the
is
correct.
it A?
Yes, which one? 3. Which one?
3. Is it A?
Okay, he's asking all are brown. Perfectly right. You're absolutely right. Well done. It's A because all are brown. Correct. Well done. Yes, guys. Good going. I have good number of students, good, alhamdulillah, good students, and I have very attractive, nice, uh, focused student and vigilant as well. Yes, which of the following crosses in which is likely to produce all blue-eyed individuals?
Thirty.
Sir, C.
C is most correct. Very good. Correctly right. Because both of them, they are recessive. So when we make a cross of this with this, and this with this with this with this, we have all the individuals, all the babies, all the children in the new generation. If father is blue eye, mother is blue eye, all the children will be with the homozygous recessive. They have blue eyes. Well done. Excellent. Excellent, guides. It's blue. Wonderful. Now we are moving to the next part. What is mutation? In this chapter after monohybrid cross. I'm sure everybody is watching the slide. When we use the word excuse me, mutation, it is a sudden change on the gene and chromosome which will cause diseases like albinism, sickle cell anemia, or cystic fibrosis, or Down syndrome, according to your syllabus. Remember. So, gene mutation, change of the gene, or chromosomal mutation, change of the chromosomes number. Take a look of this and make your concept very clear. It's very interesting, very easy. Now look at this one. This is a normal chromosome. This one is a mutant one. This one, just to make it a little ahead. This is mutant one. This is mutant two. And this is mutant three. So normal chromosome, for example, in the normal person, the normal chromosome has, this one is normal chromosome. The next one is, for example, mutation has taken place, for example, mutant 1, for example, mutant 2, and for example, this is mutant 3, for example, just to write first, it is mutant 3. Now what happens in one? Chromosome 1 has genes, for example, it has genes A, B, C, D, or E. In case of chromosome, in mutation 1, what happens? There is A, for example, there is B, there is C, there is D, and there is E. You are watching the difference, please. Remember, here is addition of gene done in mutant 1. It will result into a disease, for example, cystic fibrosis. Thick sticky mucus is produced. This type of mutation is known as gene addition. Remember, in which there is, you can see there is a duplication of gene done, and this duplication which is done at the level of B. Second, mutant 2. For example, it is gene A, gene C, gene D, gene E. So what happened here? It is gene deletion. Gene is deleted. So it is gene deletion. One gene is deleted. You have seen some people in your premises, in your school, college, and outside, around wherever you have visited. Some people are with the albino skin. They have white patches on the skin. So their skin is not producing the pigment. So their gene is deleted. Their melanin pigment is not produced. So due to gene mutation, due to the absence of gene. Third one is, take another example of mutant gene. That is known as gene substitution. So what is happening in case of substitution? A, C, D, E, and we have B. So gene has shifted from its place to some other place. This is known as gene substitution, which will result into not a normal hemoglobin, but the hemoglobin, for example, it will become a sickle shape. Or we can say sickle cell anemia. So these are the examples of excuse me, gene mutation. Take an example, take a practical example of that as well. Now look on the side, please. This is a person who is heterozygous for hemoglobin sickle cell anemia. Take a look, please. This one on the right side. HB. For example, it is HbA and HbA and HbS, for example. And here is this was the male. Now here is the female. For example, she's also HbA as well as HbS. Both are heterozygous. Externally, genotypically, phenotypically, they are looking normal, but genotypically, they are the carrier. Both the boy and the girl, both the couple, male and female. Now, when they are going to interact and they have probability, what are the chances of their children? Probability chances are as far as the normal hemoglobin is concerned, as far as the heterozygous is concerned. Now, take a look of that. Questions are based on that and they are very easy. And student, I don't know why they become feeling difficult at that time. It's interesting and easy as well. Take a look, HbA. So 25% chance is normal. Clear? Yes, everybody, yes?
Yes, sir.
Thank you. Second, what we did, just to make it clear, we just crossed this with this and wrote here. We crossed this with this and writing here. No, this is the carrier, 25% carrier. Now we are crossing with this and putting here. Take a look. It will be again HbA, HbS. So they are also carrier. So we have two carriers. This is carrier, 25% chance. This is carrier. Excuse me. 25% chance. And the last probability, which is round about 25%, that is HbS. Now, this is a person, 25% probability, male or the female, they have a disease that is sickle cell anemia. Clear? Everybody? Clear? Can you move on?
Yes, sir.
Excellent. Thank you. Now I am putting it on the group as well. Now we are moving further. Chromosomal mutation. In your syllabus, they say that Down syndrome is the example of chromosomal. Down syndrome basically on chromosome number 21. As you all know, male has 23, female has 23. So from 1 to 23, there are chromosomes, and chromosome number 21, for example, this is chromosome number 21. There is an extra chromosome. So there is an extra chromosome here. Then what will happen? The total number of the chromosome will be 24. Agree? Agree?
Yes, sir.
Thank you. So if this boy or the girl, if this male or the female, when they interact with the other, so what will happen? This 24 combined with the other normal 23, for example, their zygote, which is going to produce, definitely it will have 47 chromosomes. But that person has Down syndrome. Down syndrome person has different characteristics. Short nose, short neck, downward slopy eyes, mentally retarded, or underdeveloped brain. So these are the areas which are with this person. those who have the issue of, we can say, Down syndrome. Remember, I'm also giving it to you on your group so you can be able to go through or revise it by yourself anytime. Although recording of the lesson is also available, but right now we are moving further. Now this is about mutation. Now as we move on, there is variation. Now what is variation? Variation is basically in the characters. It can be continuous, it can be discontinuous. So there are two types of variation in your syllabus. One is continuous variation, other one is discontinuous variation. Continuous variation is controlled by many genes. Discontinuous variation is controlled by two or few genes. Remember, when we say continuous variation, it is basically controlled by many genes. This is the meaning of that. Remember, okay? And discontinuous variation is controlled by two or few genes. Example you can put here in case of continuous variation. I'm just writing in short CV, that is continuous variation. One example is height. Second example is color. Third example, you can write intelligence. Everyone has different level of intelligence. Next, fingerprints. Everyone has different fingerprints. No, these are the areas which are included in the category of continuous variation. As I told you, they are controlled by another important thing, just to add here with the same place. Many genes, controlled by many genes. Third, due to control of many genes, they show additive effect. So they show their additive effect because they are controlled by many genes. That's why. And most important thing, they are affected by environment. So they are affected by the environment. Black become white, white become black, tall become small, small become tall. These can be possible. In illiterate person become educated person even. So this is about continuous variation, and they show a graph of applied to like this. For instance, now as far as the discontinuous variation is concerned, in that case, the probability is little different, completely different. Discontinuous variation is controlled by two or few genes, and they are, as far as continuous is concerned, as I told you, height, color, shape, these are the things. Take some examples, then we move further. Height different, color different. You can see different people at different heights. All the heroes which are in the movies, which you have seen the movies, they have different heights. Plus, as you can see, in case of discontinuous variation, they are controlled by two or few genes like lobes, fixed lobe, or hanging ear lobes. Some people can roll the tongue, some people cannot roll the tongue. Some people have curly hairs, some people have straight hairs. So dominating characters, as I told you, that is roller, and recessive is non-roller. Dominating is curly hair, recessive is straight hair. So these are the areas which are, in case of discontinuous variation, which is controlled by two or few genes. Even blood group is also controlled by just four main groups: A, B, AB, and O. Now you can see in case of discontinuous variation, this is the type of graph shown. As you can see from here, some people can roll the tongue. Yes, I would like to know in my biology class. Yes, how many people can roll the tongue? Gee, Dr. Hamna, would you roll the tongue? Are you a roller or a non-roller? This is a roller. Gee, Arab. Gee, Hassan. Gee, Fahad. Anybody is a roller or a non-roller?
I am a non-roller.
Okay, I am also a non-roller. Yes. Okay, well done, no problem. And it's a recessive character, by the way. Then some people have curly hair, some people have straight hair. So curly hair is a dominant character, or straight hair is a recessive character. So some people have hanging earlobes, which is dominant character. Some people have fixed earlobes. These are some parameters relevant to continuous and discontinuous variation. I'm also putting it on your group. Now we are moving to the next last part of this chapter, which is very, very important, and that is protein synthesis. In this regard, okay. We have done this monohybrid crosses, and then we have done this. Okay, next topic which is coming, that is natural and artificial selection. It is again very important topic. Sometimes the question come, describe the role of natural selection in the development of species, new species, and describe the importance of artificial selection. So natural selection is done by nature. Remember, I am here with the your LSE LAC notes directly. Natural selection is done by nature. Remember, and in this case, there is a lot of chances of variation in the new species. So, and there is a huge reproductive potential and more adaptability, and chance of new species development is possible. But in case of artificial selection, when we say it is artificial selection, it is done by human in the plants, particularly mostly, and for the desired character, like good meat quality, or we can say a desirable feature like high milk production, disease-resistant crops. We can grow from this. For example, if wheat or rice or cotton or sugarcane, we can increase their grain. So artificial selection, if it is done within the same species, that is to increase the desired character. Or it can be done in the different species, like horse, donkey can produce mule, or lion, tiger, they are crossed and they produce liger. So these were the examples which they mentioned. Now, as we are entering into the last part of this of the syllabus, that is related to ecosystem, ecology, and biodiversity, conservation, and these areas. Quickly go through this area. Mostly the question coming on that is food chain, food web. Excuse me. Food chain, it is the interdependency of the organism upon one another. Take a look of this. For example, this is, as you can see from here, the example is given here. The tree, caterpillar, songbird, and hawk. It's mean, what is food chain mean? It is the interdependency of the organism upon one another. For example, if any one of the organism is missing, here, caterpillar is no more, then there is the food chain will finish. The songbird will die, hogs will also die. This is food chain. But in case of food web, because it is the combination of many food chain, that's why the web survived. If you take some examples, just to take you somewhere with the example to my notes, which I'm using in my classes, just to take you there as well. Food chain, food web. Everybody is watching the slide of food chain, food web.
Yes, sir.
Excellent. Now, as you can see, chain, it is the interdependency of the organism upon one another. If any one of the organism is missing, the chain will not continue. But in case of, as I told you, you can see there are many food chains are shown here. In the ecology ecosystem chapter, there are some trophic levels. There are some definitions like producers, which are the green plants. Remember, consumers are the primary consumer, secondary consumer, tertiary consumer. Remember, questions sometimes 1 marks come like that. Like this, what is consumer? So when we say consumer, it's been primary consumer, or as you know, these primary consumers are also known as herbivores. And same is the case when we go for secondary consumers. So these are the carnivores. And if we go for the tertiary consumer, these are the omnivores. So these are the 3 categories of consumer. And as you can see in the diagram shown here, we have given you some examples as well. Producer, grasses. Primary consumer, grasshopper. Secondary consumer, just to make it a little bigger, right, shown here. And also then secondary consumer, then tertiary consumer is snake, quaternary consumer, that is hawks. Remember, when we use the word decomposer, it's mean they are the bacteria and fungi. Remember, so decomposers include bacteria and fungi. So these bacteria and fungi or fungi, they are converting dead plants or animals into useful organic compounds for the plant growth. So they can ask you to define the term food chain, food web, differentiate between food chain, food web. Now, the next example we are going to go for after that, as I told you, producers, green plants, consumers, primary, secondary, tertiary consumers, herbivores, carnivores, omnivores, then herbivores, carnivores, omnivores. Then we have decomposers. These mushrooms are shown here, puffballs, and bacteria are grown. So these are the example of decomposers. Now, food web. Food web, it is the combination of many food chain. If any one of the organism is missing, the web will continue. For example, wild cat is not there, but if there is no wild cat, the web will continue because the alternatives are there. If this wild cat is not there, so alternatives for the goats, for the owls, for the kite, for the everyone have options all around. So in this way we can say, you remember a few years back there was a bird flu and people were saying don't eat chickens, and that's why. So we have other options like fish, like beef, like mutton. So just like that, if we have if there is a food chain, in this case we have multiple options, right? Now we are moving further to the next part of this chapter, that is pyramids of energy, biomass, and so there are different types of pyramids specified in your syllabi. The most interesting thing is that in case of this chapter, they are giving you a question relevant to some pyramid of energy, biomass. Sometimes they specify, describe carbon cycle, what are the stages of carbon cycle or nitrogen cycle, and what are the processes of nitrogen cycle. So we will move towards that direction. Before that, just to make a quick review of pyramid of number, pyramid of biomass. Pyramid of number means that if you look into this diagram, pyramid of number, it is the total number of individuals at each trophic level. This is the meaning of number. If we say pyramid of biomass, it is the total dry mass of the organism at each trophic level. For instance, For instance, we have corn. If this is corn, it means we have 100 corns, and on that we have cows. For example, we have 50 cows, and above that we have one human. So this is the example of, this one is the example of pyramid of number. If we go for pyramid of biomass, for example, he's showing here the total dry mass of the oak tree. It is in tons. That's why it is bigger. The trophic level size is bigger. Then caterpillars upon them are many. So shown here. Then shoes are there. Then ultimately, lastly, owl is there. So it is the total dry mass wise. Mean to say the total dry mass. Remember, so number of individuals for the count, total number of individuals. Mass, biomass means the dry mass of the, to make it a little bit more clear, it requires some explanation. I would like to clarify. Please take a look. Take a look. For instance, this is a tree. On that tree, there is, for instance, there are 10 birds. One tree, and on that tree we have 10 birds. And on the 10 birds, there are some fleas, some mosquitoes, maybe 20 mosquitoes or fleas. Now, if we want to make the pyramid of this, it will be like this. Take a look. It's very interesting. This is a tree, a tree, a tree. 10 birds and 20 fleas. This is the pyramid of number. Remember, producers, consumer, primary consumer, secondary consumer. And this is pyramid of number. Second is, if we want to make the pyramid of the same pyramid of number into pyramid of biomass, it will not be like this. One tree, 10 birds, and 20 fleas. A tree mass, remember, it's very high. That's why it will be like this. This is tree mass in tons. The same pyramid I'm discussing. Then 10 birds. 10 birds, 10 kg, for example. Look at that. Finish, khalas. And fleas, just small in grams. So this way you have to be very much careful when you are doing the or solving the question. Now, as we move on, with respect to some question relevant to that, coming about the cycles of nature. Remember, just to take you to the whiteboard for some convenience, and also showing you some. For example, there is carbon cycle. Carbon is recycled in nature by 4 main processes. One, you know, that is photosynthesis. Second is, as you know, respiration. Third, as you know, that is combustion. And fourth, as you all know, that is decomposition. So in this way, carbon is recycled in nature. They will give you a graph, a diagram, and ask you which process is taking place. So if you are clear about their concept, photosynthesis, respiration of animal and plant, combustion, burning of fossil fuel, and decomposition, dead bacteria, these dead plants and animals are decomposed by bacteria and fungi. At the end, please summarize your answer of any cycle, that all these cycles of nature, they are for maintaining the nature. So they are maintaining the nature, and also you can write that for the stability of organism. So these parameters you will sum up for all the cycles. Just to take you to the one of the question relevant to that, carbon cycle or some nitrogen cycle. Gee, everybody's here. We're watching the slide right now.
Yes, sir.
Fantastic. Now we are here on the LSE resource pack questionnaire. If you look into the questionnaire, I placed some of the question from structured question of this relevant topic. Think so. Question 1, 2, 3, 5. Yeah. It's a question here shown, showing a food web. He specified a corn, mice, grasshopper, snake, grass. So these on the bottom line, if you see, thank you. On the bottom line, if you see, so these all includes producer. So these are the producer. Remember, these are the primary consumer. Remember, and secondary consumer and tertiary consumer. Now name the trophic level in the food web is shown. State the number of trophic levels. How many trophic levels are shown here? Trophic level 4 are shown. Very good. Excellent. State the number of one organism that feed at both the third and fourth trophic level, which can eat at third and fourth. It can be mice.
Hogs.
Okay. He says that state the name of one organism that feed at both third and fourth trophic level. So if you see, mice is directly eating mice is eaten by the snakes, also eaten by the hogs. So the fourth trophic level is at the top. So feeding at both the levels done by, if you see according to the arrow you see, that is, or hogs is at the top. So top carnivores. But in case of the diagram shown here, take a look of this. Next, state the name of type of energy that transferred from the trophic level. So energy transfer from producer, primary consumer, secondary consumer, and tertiary consumer. All the energy is coming from the sun. Remember, it is one of the pyramid that all the energy is coming from the sun and goes to primary consumer, secondary consumer, tertiary consumer. Then he is specifying here. Okay, this is the next part which is coming ahead. Just to take you to the specification of this question as well. Hogs is the fourth trophic level because it can be able to eat third and second. Next is when we say the chemical. As you can see, he's asking here, the question was chemical. Name the type of energy must be transferred from trophic level. So energy is transferred, that is chemical energy, because they are eating them, and the next trophic level is also utilizing them. So after the second part or third part, the total biomass is less than, here you are going to simply specify, use the energy in your answer. These arrows, remember, these arrows are showing you energy transfer from one trophic level to the next trophic level. Remember, you will specify this thing in your answer very clearly. I'm also putting one question on the group from the past paper shown. Just to link it for a while with this. Another thing which is just to move back with the same where we were last time, because due to this scarcity of time, it is difficult to cover the last part of this chapter. Gee, now he's another important area about after this food chain, food web, or cycles of nature. One more cycle about after the carbon cycle, that is nitrogen cycle. which also I would like to share with you. I'm taking you to the whiteboard again because that is also an important cycle. Okay, we have the next page also. Look at this nitrogen cycle. Nitrogen is recycled in nature by different processes. Number one, nitrogen fixation. Number two, ammonification. Number three, nitrification. And number four, denitrification. And number five is mentioned in some of the book, that is putrefaction. Now, mostly when we say nitrification, it means that, remember, in case of nitrification, there are two processes. One from the, we can say, atmospheric nitrogen, as you know, that is 78%, and it is directly, this 78%, which is directly going to be fixed by the root nodules of the leguminous crops, legumes. They have root nodules. And they are going to fix, they have bacteria, nitrogen fixing bacteria, and they convert atmospheric nitrogen into useful compound. Second, directly from the air, you know, atmosphere has 78% nitrogen. Due to lightning sparks, light sparks, or due to thunderstorms, they directly, the nitrogen fixes with water and they produce compounds like in the soil, HNO, HNO3 or HNO2, for example. And in this way, they are going to form, we can say, these compounds are going to fix nitrogen. Okay. Next is ammonification. Ammonification basically means that all the dead plants and animals, they are acted by the soil bacteria and they convert them into ammonia compounds. This is the second way of nitrogen fixation, which is very important. It should be kept in mind that as you have seen here. Now, in case of ammonification, the dead bodies of plants, animals, these dead bodies of plants and animals, due to soil bacteria, they are going to be converted into useful nitrogen compounds. Now, what compound they are forming? They are forming usually ammonia, ammonium, NH3 or NH3 or NH4 compounds. Now, next is nitrification. Basically, nitrification is converting these compounds, which are ammonia, ammonium, which are formed from the animals, feces, dung, or by the soil bacteria due to the dead plants and animals. Now, they convert into nitrites. Remember, these ammonium compounds, they are converted into nitrites, that is NO2. Remember, in the form of NO2, and with the help of, again, soil bacteria. Inside there are specific bacteria which are in higher classes you will study, nitrosomonas, and they form NO2. And these NO2 compounds further form NO2 to NO3. That is also nitrification next step, or nitrification, we can say part of the nitrification, second part of the nitrification, and that is NO3 formation. This is also with the help of soil bacteria, Nitrosomonas, Nitrobacter. Now, these NO2 and this NO3, as I told you, nitrites and nitrates, this NO2, NO3, this is nitrification. Now, what is denitrification? basically NO3 compounds. These NO3 compounds, they are converted back into atmospheric nitrogen. Now these nitrogen compounds back to atmosphere, converting them into, we can say, you can write like this in your exam, into atmospheric conversion into atmospheric nitrogen. So this conversion into atmospheric nitrogen, you can specify in your answer. Again, it is with the help of bacteria. Names are not in the syllabus in detail, but right now you should know main parameters only. And the main parameters are the atmosphere nitrogen converts into by the root nodules into nitrogen compounds, and also by light sparks. These are the two, we can say, ways by which you can specify a nitrogen fixation is done. Second, ammonification. Dead plants, animals convert them into useful nitrate by the soil bacteria. Also, the animal feces and dung. They are also in the category of directly producing ammonia compounds. Then these ammonium compounds, with the help of soil bacteria, they form NO2s and NO3s. Remember, NO2s and NO3s compound, and these NO2, NO3s are the nitrification. Remember, denitrification, as I told you, it is basically the, you can link directly like this. It is converting nitrates, NO3, into atmospheric nitrogen. This is denitrification. Remember, it should be very much clear. As I mentioned, double arrows also. It is denitrification. No, putrefaction, basically it is different soil have bacteria, fungus, they release enzyme, and these enzyme convert soil nitrogen compound into useful nitrogen compound for the plants, animal. That is putrefaction, basically. But right now, the area which is in your syllabus, that is nitrogen fixation, ammonification, nitrification, denitrification. So these areas can be asked. If you just see the latest paper relevant to that, they have some questions asked. Mostly they are asking from that place. What is this going on? G. Now we are back here. The next important topic which is relevant to some time, how antibiotics are produced, how penicillin is produced, fermenter, and lastly, the last topic, which is genetic engineering, which I would like to discuss with you. Now, before that, antibiotic production. I would like to focus here. Take a look, please. I hope so. Everybody is watching fermenter. Please confirm everybody is watching fermenter.
Yes, sir.
Yes,
Thank-
sir.
Thank you. I'm also sending you one of the past paper relevant question, which is given in 2020. And before that also, I'm also sharing with you. Take a look of your group, as well. Have you seen the question based on the previous topic which we have recently done, and the topic which we are doing right now, they are from this. Are you watching the next question? Please take 1 minute to read that. Everybody take 1 minute to read this question, which is showing you from the gene mutation area. Second question, which we are doing right now, that is related to antibiotic production, penicillin, which I'm also putting it on the group. And recently we have just gone through nitrogen cycle, and look at the question which he has given in the past paper about nitrogen cycle. Have you seen the question on nitrogen cycle? देखो बोला तो करे at least, बंदा बताया तो करे, yes के no. Thank you. I want your participation, please. Okay, are you watching the question which I'm placing about nitrogen cycle? Please check it out. Students, are you watching the question from nitrogen cycle? Anybody?
Yes, sir.
Okay. Yeah, can you read the question quickly? Fahad, can you read the question which I have sent it on nitrogen cycle, please?
Yes, sir, sure. The nitrogen cycle makes nitrogen available for the production of proteins in plants. These plants may then be eaten by humans and the proteins digested.
Okay.
Outline the roles of bacteria in the nitrogen cycle.
Excellent. No, help nitrogen cycle, as I told you, nitrogen fixation, different bacteria are going to convert atmospheric nitrogen into in the root nodules. And then another point I specified in the previous slides that ammonification is done with the help of bacteria. Then nitrification is done. So then denitrification is done. These are going to produce nitrogen compound for the plant growth, or ultimately, according to the question, that these plants may have been taken eaten by the human and as a protein digestion. Now, when we eat that plant and that plants are eaten by the human, we can make use of that protein in our body to make bones and muscles and blood and antibodies. These are the things which are you can add in this answer, right? Now, as you move on to the next topic, which is in front of you right now, antibiotic production. Take a look. Penicillin, how it is produced. It's a very big fermenter. It is 100 to 150,000 liters capacity. And what we are going to do in it, we are adding inside a fungus penicillium cell, which we grow inside the laboratory, and ultimately we grow inside the fermenter by adding broth. We add broth in it, broth containing glucose, amino acid. We adjust the temperature by double jacket. Outside, you can see there is a cooling jacket, which is maintaining the temperature of the fermenter within the range of 24-25 degrees C. As you can see, the double jacket is shown here. At the bottom, you can see there is a air splier inside going. This air splier, which is going inside directly, we are filtering the air from here to here. We are entering the air from here to here. It is bubbled inside. So there should be a uniform distribution of air, or it can respire aerobically, and rapid growth of fungus inside that. Fungus, after 2-3 days, or we can say after a specific period of time, after getting air, proper temperature, and inside you can see there is a impaler, which is a paddle-like structure, which is going to revolve at a great speed, or we can say they are going to make a uniform or homogeneous mixture due to its revolution. So after a specific period of time, as I told you, the areas specifically in the fermenter, the broth is added. A quick review once again, just to specify this one by one. We are providing them with air inside the fermenter. We are providing fungus with air through sparger. Then stirrer is done to make a uniform solution here. This is stirrer. Sparger, fresh air is bubbled inside. Stirrer to make a uniform solution. Temperature is maintained by the cooling jacket. You can see there is an inlet pipe, there is an outlet pipe. So as the temperature is near 25, 24, 25, we open the outlet pipe to make the temperature down again. So this inlet outlet pipe is used in this way to maintain temperature. So 3 things are very clear. One, we add broth, which contain glucose, amino acid. Glucose for respiration of fungus, remember, and amino acid for the growth and development of fungus, so its proteinaceous part can be built up. Now, after a specific period of time, remember what happens. In the fermenter, penicillium fungus, it produces excretory waste material. This excretory metabolic waste material which is produced, that is known as penicillin. Remember, it is extracted, purified, and ultimately it is going to be separated after its purification into the purification chamber in this way. I'm putting another question from the past paper directly. Please take a look relevant to this topic. I would like someone to quickly go through how the student are writing this answer. Ji, are you watching the question? It is on air related to fermenter.
Sir, only the screen with notes is visible.
Okay, I think so. It is on air now, and it is here on the group. Please check it out. Question is, describe large-scale production of antibiotic. Explain the importance of each stage in this process. Ji, have you seen the question? Yes, guys, are you watching the question from the past paper of
Yes, sir.
fermenter? Thank you. Ji, Hamna, I would like you to please participate here to go through this question, please. Yes, your participation is highly welcome. Please go ahead. Yes, Hamna is here. Yes, Hamna is here. Okay, no issue if there is mic issue. No problem. Arib is here with us. Arib, would you like to go through the question, please? Arab Zahra.
Sir, I can't see it.
You can't see the question? Have you? The question is not
No.
on the group. Oh my God. But students, it is on the group. Please check it out. It is on the group. Okay.
Oh, my net is slow.
Okay. Just a minute. I put this question in front of the slide as well. No need to worry. By the way, question is, describe the large-scale production of named antibiotic and explain the importance of each stage. Now here is this question, please. Ji, Arab, are you watching the question right now? It is in front of you on the slide, on the screen. Everybody is watching the question, please, from the past paper.
Sir, it is
Sir,
not
it
visible.
still shows the slide of red banking and all.
Excuse me, I'm saying that it is on the screen. Please check the screen now.
Yes, I do see it now.
Excellent. Yes, go ahead. Arab, read the question and the answer. Try to go through it.
Describe the large-scale production of a named antibiotic and explain the importance of each stage of the process.
Okay.
Penicillin is a large-scale produced antibiotic. It is sterilized inside the fermenter to prevent contamination. It helps with respiration, and protein amino acid helps with its growth. Oxygen is provided for aerobic respiration to take place. The
To take place, and the sparger helps in the increase of the surface area, particularly the paddles are here, which is going to make a stirrer inside to make a uniform solution. Temperature is balanced, is going to be controlled by the cooling jacket 25, down to about 25 to even 45, 40 approximately. And pH is also maintained between 5, 6, 7, 8. The temperature kept within this range and pH kept within this range because it is optimum for the growth of fungus. Remember, student, this is very important area. And so that maximum growth of fungus can take place, glucose is utilized for the growth of fungus, and amino acid is used for the growth and development. Glucose for respiration, remember, and amino acid for the growth and development, for DNA, RNA, protein formation of the fungus. At the end, please last two lines, through filtration and crystallization, then the purification is done. So extraction, filtration, purification, crystallization of antibiotic penicillin is done in the form of tablets, injection available in the market. Remember, this is a question which is asked sometime about other things as well, like single cell protein. So only that difference, the difference is the difference of fusarium fungus. Here is the penicillium fungus. Now we are back here, moving to the main topic which we were discussing. After the antibiotic is genetic engineering. Genetic engineering basically is the latest science which is prevailing all around nowadays, and it tells us how the genes are transferred from one organism into another organism basically. So this is our last important topic which I would like to share with you right now, and with some question at the end. So genetic engineering, basically it is transfer of gene from one organism into another organism. For instance, what we do here in this case, the genes are taken from the human pancreas cell, and which synthesize insulin into the bacteria cell. So for that purpose, we are using, definitely we cannot use scissors to cut the chromosome. So we use different enzymes. So for that purpose, try to understand, it's very simple. Take a look. For example, there is a test tube. This is a test tube, and this is another test tube, right? In this test tube, we add bacteria, or what we add inside? We add inside this test tube a bacterial plasmid. Remember, we have another test tube. Just to make it easy for your convenience, I'm doing like this. The insulin synthesizing cell from the pancreas we take, and we remove from that cell the chromosome part or gene part of the chromosome which synthesize insulin. I'm just highlighting the chromosome. It's very bigger shown here right now, just to make it little smaller as well. For example, this is the chromosome piece shown here, and all this chromosome which is shown here inside, this small piece we are going to remove out, or we were going to cut out. This is a bacterial plasmid all the way, and this bacterial plasmid, its small piece, it is going to, we want to cut it basically. We want to basically cut it. So for that two purposes, remember, to cut this plasmid of the bacteria or to remove this piece. I would like to take you on my slide. It will be better. It will be more easier too, because I don't place any diagram here. It is on my slides. It will be more convenient to take a look of that. Everybody is watching my slides right now?
Yes, sir.
Fantastic. Now, in case of genetic engineering, as we are continuously saying, what we are doing basically, we are removing the insulin gene out from the human pancreas cell. Second, we are cutting the bacteria plasmid. So these things which we are going to do with the help of enzyme, we are going to do that. Now take a look of this diagram which is in front of you. I hope so you're watching the slide. Human cell you are watching on the left side, bacteria cell you are watching on the right side. Everybody is watching that?
Yes, sir.
Excellent. Now, with the help of enzyme endonuclease, we are going to cut open the bacterial plasmid. First step here. If you look at that, these are the plasmid. We isolate it and we cut it from here with the help of enzyme endonuclease. Secondly, we also remove the also human insulin. We also remove from this place just to make a, what is this? Okay, I will share this, no problem. Just to take a look of this one, this small piece of human insulin synthesizing gene area is removed with the help of enzyme endonuclease. Second, we cut open the bacterial plasmid as shown here. You can see with the help of, again, the same enzyme we use, and that is endonuclease. Remember, its name is endonuclease. So endonuclease. Now what we do after that, we fix human insulin gene into the bacterial plasmid, and this is done with the help of another enzyme that is known as DNA ligase. So this is fixing the human insulin into the bacterial DNA. Now we put this DNA, recombinant DNA, this is known as recombinant DNA now. We put this recombinant DNA into the bacteria again, because it multiplies very fast inside the bacteria, right? So it will multiply very fast inside the bacteria, remember. And within short time, we have millions of bacterial recombinant plasmid inside, or we can say recombinant plasmid inside the recombinant bacteria, we can say inside. And then from there, we can easily purify or extract this insulin gene and going to convert them or pack them in the form of injections or tablets or digits which are available in the market. So this is about the way by which the genetically engineered insulin is produced. There are some issues they mentioned. What are the advantages? What are the disadvantages? No doubt, it directly, quickly converts glucose into glycogen. So it is compatible. It is not highly expensive. It is cheaper. Anyone can easily purchase that. So it is matching with our insulin as well, as far as the bacteria insulin is concerned. Plus, you can also add this thing that because it is readily available, matching with our insulin as well, or quick response, rapid response, glucose is converted to glycogen. These parameters you can be able to see here. What we do, cell-containing genes that codes for human insulin, then DNA is extracted, introduced into the bacteria. These bacteria grown gene inside, and ultimately one bacterium containing the gene for insulin is identified. Then we finally, when we put plasmid in the bacteria, we grow that all recombinant plasmid and the bacteria inside the fermenter. Remember, this is large scale production, as you have just gone through fermenter in the previous topic. Then this fermenter, we make large scale production of human insulin, which is at the end purified and extracted and again sold in the market. So as far as some of the questions some students said about to share this image, just to take this image as well. Although my brief notes are although on the group as well. One question I also placed from this topic. Okay. No, this is about the question relevant to. Now, sometimes they are asking about genetically modified crops. What are their advantages? What are their disadvantages at the end? So you can sum up this area by advantages of these crops are definitely they are giving us high yield. They are drought resistant and less water requiring, and their growing season is shorter, and their yield is high. They are disease resistant, and these are the area which are related to genetically modified crops. Even genetically modified, we can say crops, and even we are using in the cows different hormones we are using to culture or to increase their milk production in the cows, right? There are some parameters which are at the end related to. I would like to quickly go through that as well, because they are sometimes asked: pollution types, acid rain, conservation of species. On that, there is a question which was in the paper about when we say conservation. Conservation basically it is the preservation and protection of resources, whatever we have. We have different types. Definitely, we want to conserve our animal, our plants. Otherwise, they are going to be extinct. So proper management should be done, proper control should be done, proper organization should be done for that. So in this way, we can conserve our resources, even it is related to animals or plants, or some of the species which are in the natural resources, we can say, other than water and fuel, they are related to like endangered species, because we are continuously migrating from rural area to urban areas, and deforestation is done continuously, urbanization is done continuously. So species are going to be too much in a situation of endanger. Their number is decreased. So for that purpose, you can specify zoos can be formed, or seed banks can be produced. So we can grow different types of seed crops in that banks. We can produce different types of crops in that centers, or we can make storage of different seeds in the seed banks. So whenever they are available, they required, they can be available. So in this way, we can preserve that. And we can make more and more forest area, reforestation can be done, or genetic techniques or genetic technology can be done to improve the crop plots, or also the animal, we can say, quality with respect to wool quality. These areas we can also do. Now, as far as the last point of this conservation is concerned, that is endangered species. As you know, this thing that there is a lot of destruction in the environment because of industrialization, pollution. and climatic changes. So species are animal species, plant species, they are also in a situation of endanger, or they are going to be extinct. So we have to provide the environment which should be free, we can say a pollution free environment, or such solution should be done which can be able to control them in such a way that we can be able to control our we can be able to manage our ecological system or balance the stability of nature by making such resources. So in this regard, if you go through my notes, they are very much precise in this LAC pack. My detailed notes are on the resource pack available. Students, main areas which I tried my best to compile in the whole syllabus, relevant to ecology, relevant to reproduction, relevant to inheritance, and other topics of previous syllabus, coordination response, then systems of the body, circulatory systems, and digestive system, excretory system, and plant section, food test, and enzymes up to the beginning. I hope so. We have covered maximum areas and past paper question to relevant within this period of time. If you will simply go through the latest 2-3 years paper, which are, if you have available, definitely, and if you want to discuss, so for that purpose you can directly take my number from the LAC platform, and you can discuss that areas. Plus right now I would like to get something from your side, that in this whole yesterday's session, in today's session, I tried my level best to compile all the syllabus, but I am telling you, you people really participated very well and gave a very quick responses. Any thing would you like to share? Would you like to ask here? Because I'm compiling right now. Ji, your feedback from the point of view of this regard. Ji, Dr. Hassam is here with us. Dr. Hamna is here with us. Dr. Fahad is here with us. And Arif Zahra is here with us. Ji, Arif Zahra, Fahad, Hamna, and all the people, I would like you to please go through the maximum latest past paper, right? Everybody? Clear? Are you getting the voice?
Yes, sir.
Yes, sir.
Thank you. Thank you, guys. And I wish you all the best for your paper. And if you have any query, you can put it on the LSE and you can get my number and you can discuss with me. You can use my resource pack. And inshallah, I will try my best to help you a lot in this matter. And my prayers are with you and with all of you people. And also, remember, ask your parents or particularly your mother to pray for you, because mother prayers are very much stronger as compared to everyone. Thank you very much, guys. Have a nice day and wish you all the best for your paper.
Allah Hafiz, sir.
Allah Hafiz. Allah Hafiz, everybody. Please don't forget to mention your feedback to the LSE platform. I shall be very grateful. Thank you so much, everybody. Wish you all the best. Allah Hafiz. Allah Hafiz. Ji, anything else? Thank you very much, everybody. Allah Hafiz once again. Fahad, any question? Allah Hafiz.